Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's guest is Sean McClain. He's the founder and CEO of Vancouver, Washington-based Absci. Sean started the company on his own in his early 20s, straight out of the University of Arizona, with the encouragement of his dad. You could call it a scrappy garage biotech with no traditional VC backers at first. He found a way to do something useful, developing an E. coli assay to test antibodies quickly and cheaply against molecular targets of interest to biopharmaceutical industry customers. The business soon evolved, and AI emerged. One of the key questions 5 or so years ago was how to feed AI with relevant data sets from wet labs, so it would have the kind of quality data that's the essential grist for AI to learn from. Absci embraced the new tool with an eye toward using it to speed up discovery of biologic drug candidates. It decided to discover and develop its own. Absci is now a public company. It has 200 employees. McClain, as the founder-CEO, has raised more than $530 million. The company is preparing to take its first antibody drug candidate, aimed at the TL1A target for inflammatory bowel disease, into clinical testing in 2025. This would not be a first-in-class medicine. There are 2 other notable antibody programs against TL1A that are further ahead in development and which were recently acquired by Merck and Roche. The AI drug discovery wave has had some big ups and downs over the last few years. I think McClain is one of the entrepreneurs out there seeking to strike a balance between the optimistic sense of what's possible, tempered with the knowledge of how little we still know about biology and how far AI still has to go in drug discovery. This was a conversation that I think is both uplifting and grounding at the same time, if that makes any sense. Now, before we get started, if you like listening to The Long Run, you'll love a subscription to Timmerman Report. This is where you can read my coverage of the most interesting startups in biotech, my weekly Front Points column, and commentary from a rotating cast of contributing writers. Individual subscriptions are available on a monthly, quarterly, or annual basis. Group subscriptions are available at a discount. Go to timmermanreport.com and click on subscribe for more. Now please join me and Sean McClain on The Long Run. Sean McClain, welcome to The Long Run.
Thank you so much for having me here, Luke.
So, Sean, it's great to see you here in person at the Bio International Convention in San Diego. Lots of interesting people working on interesting things, including you and your company. Really happy to have you.
Yeah, it's great to be here at Bio, and we were just chatting beforehand. It's been a rough couple of years, but the resilience of this industry really amazes me, and the ability to continue to drive innovation in difficult times. And ultimately, we're always reminded that patients come first, and even in those hard times, patients and the therapies we can get to them are what keep you going at the end of the day. And so it's just a great reminder being here at Bio.
Yeah. And at the end of this, you come out with a medicine, then we can compare our battle scars.
Absolutely.
Okay. Well, let's start from the beginning with your story. Where are you from?
I am from Portland, Oregon, actually just outside of Portland in Sherwood, Oregon, which has now become an amazing place for Pinot Noirs, the Willamette Valley.
Okay.
So just outside of there is Sherwood, Oregon.
And what did your parents do when you were a kid?
Absolutely. So my dad was in finance. He got a CPA degree from the University of Oregon and went on to work at Ernst & Whinney, which is now EY, and was doing tax returns, and realized that he didn't want to continue to do that. He really loved coaching. And, one of the areas and kind of the opportunity he got was to become a general manager at this circuit board assembly company. And, he actually had the opportunity at one point in time to acquire that company, and he ended up not doing it. It was like one of the biggest regrets of his life. And it was one of those things that at a young age, he always reminded me of the ability to kind of chart your own destiny, to make a difference in the world, to, become an entrepreneur. And my dad, went on to become the COO and president of the circuit board assembly company. And one of the things that I just really learned from him was that, that leadership, that ability to coach a team and bring somebody, bring a team together to go and accomplish, really extraordinary things that you wouldn't think were possible. And then my mom was more the creative of the family. She was an interior design, or she had her own interior design company. And I think that's really kind of actually what spurred my love for entrepreneurship and science. And, that creativity is, I think, really baked into me and what I do and the science. And I definitely got that from my mom, and definitely kind of that entrepreneurial drive definitely came from my dad.
I love to hear you say that about creativity in science, because some people don't get that impression about science. They think it's sort of memorizing of facts and the Krebs cycle and all this stuff. But actually, this is a creative field. You have to imagine things and then gather all your data and run through the scientific method. It is closely related to art in some ways.
100%. And I firmly believe that in order to create a therapy for patients, you actually have to believe that it's possible. And it may be a 0.01% chance that it works out, but if you don't believe in it from the get-go, it's never going to ultimately come to fruition. And there's a lot of then creativity that goes into actually creating that therapy that's ultimately going to make a difference in patients' lives.
Yeah. At the beginning, you don't have all the data.
Absolutely. Yes. You have a dream.
Right. Then you collect the data.
Absolutely.
Okay. So your dad instilled that drive, that value around entrepreneurship. Yeah. Starting your own business, taking that risk, exhorting the troops, all of that.
Yeah. He actually got me to start my own lawn mowing business when I was probably 10 or 11. I wanted a video game during the summer, and my dad told me, he's like, well, you need to go get a job. I was like, Dad, I'm 11. No one's going to hire me to work. That's child labor. And he's like, well, it looks like your neighbors need their lawn mowed. He's like, why don't you go ask them if you can mow their lawn? And I was an extremely introverted kid, and that totally terrified me, going and knocking on a door and asking our neighbors if I could mow their lawn. And anyways, I ended up getting enough courage to go knock on the door and asked if I could mow their lawn. And they looked at me as this cute little kid and was like, absolutely, here's $10 a week, you can mow my lawn. And ended up that whole summer mowing their lawn, and I got so much joy out of that. And, I ended up actually not even buying the video game that I wanted. I was actually thinking about the next summer. How could I go and, grow from, one customer to 10 customers? And so that next summer I went out, I had my little business card, Mowing Express, and I went door to door and ended up, growing it to a pretty substantial business that paid for most of school. The biggest lesson I actually learned from that business was the ability to go and sell it. I actually should have went and sold the customer list to the local lawn mowing company, but I missed my first opportunity to actually sell a company. But that, even as a simple business like lawn mowing, it taught me how to be, courageous and to have conversations with people and put yourself, outside of your comfort zone, and created that drive to go and build something, and even as simple as a lawn mowing business. And it just gave me a lot of joy.
Well, you said you were an introverted kid, and that forces you, at least for a while, into a more extroverted mode.
Absolutely.
Okay. So what kinds of things did you study, and were you drawn to in school?
I loved math. Math was a big component of my life. And, that kind of gave me a love for physics, and ended up, in high school, had a big fascination with biology, but I never thought that I was actually going to go into bioscience and life sciences. It was really kind of this idea of, I want to become, a mechanical engineer. And so ended up going to the University of Arizona, and, was going to, ultimately either become a chemie or a mechanical engineer, and ended up taking a biochemistry class.
What drew you to the University of Arizona?
Ultimately, I grew up in the Northwest, and I had the opportunity to either go to UW or University of Arizona. And it was, again, the putting myself outside of my comfort zone of kind of leaving home, doing something new. Pacific Northwest, I knew. And it was like, let's just go to the desert, and they have a great engineering program, and let's go try something new. But again, kind of putting myself in an uncomfortable situation. None of my friends from school were going there, and it was good academics for engineering, and I also just wanted to get out of the rain and into the sun as well, and ended up choosing the University of Arizona.
So you go there and you're thinking about engineering, and how did things veer toward biology?
Yeah, so I ended up taking a chemistry class. It was actually Dr. Pollard at the University of Arizona, and his ability to talk about chemistry and the applications, and one of the applications was biology. It really got me fascinated in this idea of engineering, biology, and then I took this biochemistry class, and that really instilled in me this idea of like, wow, you can actually apply engineering principles to biology, to living organisms. And that was right around the time the term synthetic biology was being coined. And it kind of drove me into this obsession of again, engineering living organisms. And if you looked at the history of biotech, it began with Genentech. And Genentech was wanting to take insulin that was derived from pig pancreas and produce it recombinantly in E. coli. And they figured out a way to engineer a very simple organism, a bacterial cell, to produce insulin. And the industry ended up then shifting from producing insulin in E. coli to monoclonal antibody-based therapies. And Genentech actually back in the day tried to produce antibodies in E. coli, but it just wasn't capable of producing it. So they switched to producing these in mammalian cells or CHO cells. Mm-hmm. And so kind of the original idea I actually had for Absci was engineering E. coli to be more mammalian-like, to produce antibodies. And it was kind of, what's the significance of this? Well, by making kind of that switch from a simple organism like E. coli to a more complex organism like mammalian cells, the development times, dramatically increase, and the overall costs increase. So it was this idea of like, well, if you could produce it in that simple organism, you could lower the costs for manufacturing of these, biologic-based therapies.
This is sort of that engineering mindset coming through.
Absolutely.
You saw that, okay, there's a better, faster, more efficient way to do things, even with biology.
Absolutely. As complex as biology is, it still is based in physics. It's based in engineering, principles. It's obviously the most complex system that's out there that we know, and there's still so much to understand, but ultimately you can engineer biology.
But this was, okay, so to build off that Genentech analogy, you had a gene sequence, we knew what it was for making insulin, and you could take that sequence and put it into the nucleus of a mammalian cell or a bacterial cell, in this case E. coli, give it some nourishment for a little while, and then watch the protein come out and purify it, and golly, you've got a drug that you can inject into people to treat their insulin, to treat their diabetes.
Absolutely, which is incredible. Mm-hmm. Absolutely incredible. You literally engineered an E. coli that does not produce insulin to produce insulin that can then treat all these patients that have diabetes. That's mind blowing.
And your undergraduate mind was captivated.
Absolutely.
Okay, so you graduate from Arizona in what year?
2011.
Okay, so as you said, this was the beginnings of synthetic biology. I think Drew Endy, Stanford would have been one of the early movers there. There were others.
Drew Endy was really big, and that's, I think, a couple of years after that is when Ginkgo Bioworks was founded, and I mean, obviously, you had some of the other synthetic biology players that were in the space, like Amyris. A lot of them were focused on synthetic biology from the kind of biomaterials and energy space, but not as much focused on the therapeutics side of things.
Mm-hmm, mm-hmm. So you graduate 2011, and what were you thinking that you would do?
Absolutely. So I was looking to go get my MD PhD. I switched my majors. They didn't have a bioengineering degree at the University of Arizona, so I ended up getting a degree in molecular biology and chemistry. And it was this idea, this fascination again with going back to engineering E. coli. It's like, okay, if we could engineer E. coli to make insulin, we can engineer E. coli to make antibodies. And I had the opportunity to go get my MD PhD, but it was like this nagging feeling of like, I need to see this through. And so I actually talked to my dad. I was like, look, I'm thinking of just graduating a year early from school and pursuing this idea. And what are your thoughts? And my dad was like, you should go do it. You should go see if this idea has legs. And I remember he's like, what do you need to do? Like, how do we go get this started? I was like, well, we need to go find lab space and we need to go find equipment. And I remember sitting with him at the kitchen table and going on a site, Dove Bid, which, sold surplus lab equipment from like the 1980s, and sitting there and, going and buying all this used lab equipment to go engineer some E. coli to produce antibodies. And ended up finding this basement lab in Portland, Oregon. It was like this 200 square foot basement lab that looked out into a garage. It had wooden lab benches all along the wall and a garage sink. And we ended up getting all this surplus lab equipment into this this small 200 square foot basement lab, and ended up spending the next 18 to 24 months engineering the E. coli to produce some antibodies.
Not a lot of parents would have been so encouraging when you could have gone to MD PhD school.
Absolutely. Yeah. I asked my dad, a little bit ago, like, why did you, support me in this? And he's like, look, ultimately, if Absci failed, it was going to be a great education for you. He's like, embrace the failures, Sean. He's like, you know that all the failures along the way with Absci, you've been able to learn and grow from it. And so whether or not Absci was going to be a success, that really didn't matter. This wasn't a very important lesson for you to learn. And for you, he's like, I didn't want you to be in the same situation that I was in, regretting not taking that leap of faith and going and starting a company. And again, he's like, I had no idea if it was going to be successful or not, but I just figured it'd be another, couple years of education for you, and you could easily go right back into school if you wanted to.
Sure. And so now this is 2011. So this was also during the Great Recession.
Yes.
Not a lot of companies were getting started or raising the big venture capital amounts that they had 5 years previous. There was a lot of loose talent and equipment to be had. How much did this cost you to get this? Did you buy this stuff off, the proverbial garage sale?
Yeah, it was $40,000. That ended up getting me the equipment that was needed to get the company off the ground. And I had zero connections into the industry, completely naive coming into all of this. And so what did I do? I put on my door-to-door salesman hat, and I literally went on to the top 100 large pharma websites and found the BD contacts, and we're literally just cold emailing these BD contacts saying, hey, just started this company. We're looking to produce antibodies and E. coli. We believe that this will help lower the manufacturing costs and the development times. Is this a technology that you're interested in? And we ended up having 3 large pharma reach back and engage and said, yeah, we're actually interested. And one of them gave us the kind of the specs that they wanted to see to engage in like a feasibility study or an initial partnership, and that was pure gold. Like that allowed us to be able to essentially have the blueprints to build out this technology. And so we spent that 18 to 24 months building the technology to the point where we could go back with that initial data. And lo and behold, we were able to engineer the E. coli to produce antibodies, built an initial data set, took it back to all 3 of those large pharma companies. And the one that had kind of given us the most information, the blueprint that was really engaged, they ended up saying, this looks great. Let's enter into a kind of initial proof of concept study. And I remember they had given us 5 proteins that they wanted us to produce in E. coli to see the breadth of it. It kind of represented the 5 therapeutic modalities that they were most interested in. So it was Fc fusion, full length antibody, a Fab, an Fc fusion, bovine growth hormone, and insulin. I think I said Fc fusion twice. Mm-hmm. I remember that, I given the price, and they're like, oh, well, we can only afford to do a proof of concept on 1. I was like, you know what? Let's just do all 5 for the price of 1, and ended up being successful with all 5 of those. And they ended up giving us an acquisition offer at that point in time.
Well, so this would have been your first real revenue.
Yes.
As a company.
Absolutely.
24 months in.
Yes.
So you had been working on your platform in the basement.
Yes.
This 200 square foot lab by yourself.
Yep.
Can you say a little bit about what you found attractive or appealing about E. coli cells compared with mammalian cells, and like what you really thought you might be able to offer that was different and better to the pharmaceutical industry?
Absolutely. So, producing antibodies or any kind of complex mammalian protein in E. coli, most of the time what ends up happening is these proteins aggregate because they don't have the post-translational modifications. And so we ended up engineering the cells to be able to enable these post-translational modifications to happen. And so one of the post-translational modifications you need is disulfide bonds. And so what we ended up doing was we engineered the cytoplasm of the E. coli to be what we call semi-oxidized, where it's oxidized enough to form disulfide bonds, but it is reduced enough to be able to grow to high cell densities. And that was one of the kind of the core innovations. And then the additional one was being able to control the transcription and translation. So how quickly, a protein is folding. And so we were able to kind of engineer in some inducer, to be able to essentially control the rate of, transcription. And then additionally, we knew that, every protein kind of has its own personality. And so it needs different, folding and expression solutions to fold it in the proper conformation to, be soluble and to be active. And so we kind of took all of these components and were able to engineer that into an E. coli and be able to produce an antibody that mimicked that of a mammalian derived antibody. Now, the one post-translational modification that we did not have was glycosylation. But one of the reasons why we didn't end up engineering that in was because Genentech had done some studies showing that the PK/PD profile of a glycosylated antibody was very similar to that of a glycosylated antibody. And a lot of at that point in time, and still now, a lot of the focus was on IgG4s, these neutralizing antibodies where you don't want effector function, and an aglycosylated antibody doesn't have effector function. And so that was kind of the the niche that we were focused in on, as well as kind of these next generation modalities that were coming out that didn't need glycosylation, like Fabs, Fc fusions, these T cell engagers, and those were actually difficult to produce in mammalian cells as well. And that ended up kind of being our niche, was these kind of next generation biologics being produced in E. coli.
And of course, E. coli, it's a fast dividing cell. It's cheap.
Absolutely.
So if you are the pharmaceutical industry and you want to test some basic concepts, this is a pretty good place to get some quick and dirty data that you can then replicate in mammalian and cell lines, and also, play around with things like glycosylation, figure out how important that really is.
Absolutely. I mean, you were able, we were actually able to model, some of this out. I mean, depending on the protein, you could get, COGS reductions of, 25 to 50% for the actual drug substance. And, with the manufacturing times, we're able to show that, you could probably decrease it by, 50%. And so, at the time, like that's, especially the timelines, those were really exciting, metrics that I think got, some of these initial pharma partners excited.
Okay, so you got your first customer who's paying you real money and you're feeling pretty good about yourself, and then they offer to acquire you?
Yes.
For how much?
I can't disclose that. It was enough that my dad was like, "You're crazy for not taking this offer."
And how old were you at this point?
So when I started it, I was 21. So I was like 23 or 24 years old at this point.
Oh, so did you have dreams of sports cars and this kind of stuff?
No, it was funny. I had dreams of continuing to build Absci. I wasn't ready to sell. Like I knew that this could have an impact for the industry, and I didn't want it just to have an impact for, X Farm. I wanted to have an impact on the whole industry. And it was like, there was so much left to build and so much more of an impact to have. And so it wasn't about sports cars. It was about building a company that was going to have an impact on patients' lives. And so I ended up turning that down. But before I turned it down, I went and shopped that to all the VCs in the Bay Area, and ended up getting some term sheets for our Series A financing, and ended up the round ended up being led by Phoenix Venture Partners in the Bay Area, and Zach Jonasson, who and then one of the big reasons I ended up going with Phoenix Venture Partners was the relationship that I had with the general partner at Phoenix Venture Partners, who was Zach Jonasson, who him and I had really connected, and I felt like I could learn what it meant to be a venture-backed CEO from him. I knew that he had the best interest in not only Absci and like the financial returns that it could have for Phoenix Venture Partners, but he also had an interest in me and investing the time into me to ultimately learn what it meant to become an actual CEO and to grow a biotech company. And so that was that was a really exciting moment for me to be able to raise, the first venture round at Absci.
What do you think he saw in you? Because you didn't have sort of the classic pedigree of venture-backed CEO. Usually you have to be 40 years old and, have gone to Harvard or Stanford or whatever.
Absolutely.
You went down there and made your case and were able to get at least some backing.
Absolutely. I mean, look, you're exactly right. You're exactly right. Like, I shouldn't have gotten the financing, the VC financing that I did. I didn't have the pedigree. I didn't have the experience. And I think what he saw was that desire to build a great company, and that, I had this like determination, this drive. And, I think that, I call it the ability to will something into existence. And the sheer tenacity, to, continue to build something. I think he saw like, wow, okay, this kid graduated, a year early from the University of Arizona. Within a couple, of years, he's got an acquisition offer from a large pharma. Like, he must be doing something, right. And I think that he kind of just saw that, desire and ultimately the passion that I had. And it was, I call it, grit. Angela Duckworth's, book Grit. I mean, it's she says, grit is a combination of passion and perseverance, and I think that's ultimately what he saw in me.
Key ingredients in entrepreneurship.
Yeah.
Okay, so you raise your fund or your first round, and then you go to work on building out the company. How did it progress over the next few years?
Yeah, absolutely. So, one of the kind of the key things that we, saw was the industry was going into these new novel modalities. And actually the interest from pharma wasn't so much on the antibodies, it was on these next gen biologics, the Fc fusions, the T cell engagers, the Fabs, kind of these new kind of crazy, protein fusions that were coming out. And these were difficult for both biotech and large pharma to make. And what we ended up seeing with each of these was that there wasn't like one cell line we engineered that was perfect for, all of these different modalities. We'd like to say like every protein has its own personality. And so we needed to figure out like the right folding and expression solutions for, every single new modality that came through. And one of the early scientists that we brought on, who's still with us today, phenomenal scientist, Jalu. He came from Gilead, and prior to that, Pfizer. And he was this assay guru, and he joined when I think he's employee 11 or 12. And so took a huge bet on us when we were super early. And, he figured out how to engineer this screening platform where we could, screen, millions of E. coli cells that were producing, different chaperones or these different, folding and expression solutions, and basically be able to kind of find that needle in the haystack, that solution that was going to give you the highest yield and the highest quality protein. And that's what we ended up calling our ACE assay. And so it essentially kind of turned into this, turnkey protein expression platform where, in a very rapid manner, you could find all of the key ingredients that were important for making and producing a particular protein of interest at high titer and high quality. And so that's where, we ended up, bringing on that technology. And, we ended up, honestly, kind of by accident, ended up getting into drug discovery. We ultimately weren't focused on drug discovery, and we were working on this program with a publicly traded biotech company.
So these companies would come to you with, here's the target. We want to make some kind of targeted antibody or exotic biologic against it. Can you make some of these?
Yeah.
Against our target?
Yeah, absolutely.
And then it's ours.
Yeah, absolutely.
So it's fee for service.
We were charging milestones and royalties as well. And we were able to get milestones and royalties because in a lot of cases, the protein was produced at such a low level that it wasn't manufacturable. So if we weren't able to produce it at high enough titer and quality, that program wouldn't advance into the clinic. And so there was, a lot of value that the platform was adding. So we were able to justify to kind of pharma and biotech that, we should, share in some of the success that this drug, has, because, we're helping together get it into the clinic. And so we would charge a fee, and then we get milestones and royalties for every kind of, molecule we worked on.
And you'd come up with a process to help scale it up?
Exactly. So then we had, partnerships with, CDMOs that would then, scale this up for our partners. Like we had partnerships as well as investments from, companies like AGC, KBI, as well as Danaher. And, and ultimately we would then tech transfer, the process over to them to then do the scale up and the actual manufacturing itself. So we were this technology provider for, protein expression.
If you like listening to The Long Run, you'll love a subscription to Timmerman Report. This is where you can read my coverage of the most interesting startups in biotech, my weekly Front Points column, and commentary from a rotating cast of contributing writers. Individual subscriptions are available on a monthly, quarterly, or annual basis. Group subscriptions are available at a discount. Go to timmermanreport.com and click on subscribe for more. So how did you end up making the switch into the more high-risk, high-reward, speculative arena of drug discovery on your own?
Absolutely. So again, we ended up kind of getting into it by sheer luck. So we were working with, I'm sure that you've heard of this company, Phase Bio. They were, working essentially on this antidote. They were, had a fab that could bind to a blood thinner, and essentially for emergency use, if you had a heart attack or stroke and you were on this blood thinner and you need to go into open heart surgery, there you could basically have this fab bind to that blood thinner to basically render it inactive, so you could go into open heart surgery. And the dosing on this was extremely high, and so they needed to increase the overall titer of this compound in order to make it economically viable. And so we ended up working on this program, and we took it through the ACE assay, and we were increasing, the titers on it, hitting the key milestones that the partner wanted. And then we ended up running into the scenario where the ACE assay was reading out this higher titer or higher yield cell line. But then when we went and purified it, it was the same titer as everything else. We didn't see the increase, and so we reran it again. ACE assay is reading out higher titer, but then when we purified it, it's not. We're like, what's going on? Well, then we ended up sequencing the plasmid, and there's a point mutation. And what we ended up seeing was that that point mutation, when we purified it, it wasn't giving a higher titer. It was actually giving us higher affinity. And it was like, oh my gosh, this assay not only can look for titer, it can also look at affinity. And that was kind of a big breakthrough, because getting affinity data is very laborious process, where you have to produce it in, produce the antibodies in mammalian cells, and then, screen it by, with SPR or BLI, and you can maybe only screen like 1,000, antibodies in a given week. Well, with this assay, we were able to show that you could, if you correlated the kind of the ACE affinity with, kind of gold standard like SPR and BLI, you had a correlation of 0.9. But the throughput was, instead of 1,000, you could look at, millions. And it was like, oh my gosh, we could, rapidly use this in drug development. So you could essentially, use it to hone in the affinity you wanted, but also the titer. And so you're kind of doing this drug discovery and cell line development simultaneously. And you could utilize this for kind of next generation biologic. So that's what then got us into drug discovery.
You said we can discover our own high affinity biologics.
Yeah.
And these high affinity, by that you mean the ability to bind tightly with a
exactly.
target.
Yep, exactly.
To hit what you want and hold on to it.
Absolutely.
At least for long enough
Yeah.
to get the kind of action on that disease cell that you want.
Absolutely.
And that's actually been a weakness of many previous biologics, where they bind, but they don't hold very tight. They bounce off.
Absolutely.
Mm-hmm.
Yeah. And it was really interesting, because like the data that we were just getting out on this was like orders of magnitude more than anybody else in the industry. And another kind of point in time where we got lucky and we capitalized on our luck was when transformers came out in Google, kind of the first generative AI models. I was in 2018. Google came out with these transformers.
Was this around the same time that you had the epiphany about drug discovery?
Yes, this was
2018.
2018. And it was like, oh my gosh, like if you could take this affinity based data with these transformers, with these Gen AI models, you could really go from this paradigm of this trial and error process, where you're searching for a needle in the haystack, to actually being able to create the needle from scratch with all the specifications that you wanted. Because again, with drug discovery as it exists, you go through like an immunization campaign. So you immunize let's say a mouse or a rat with your target or antigen of interest, and then the animal uses its immune system to generate antibodies. But you have no control over where the antibody binds, the epitope, and you have no ability to control the affinity. And if you could, train these, if you could take all this data, train these, Gen AI models, you could start to explore a much larger search space, and then in silico be able to design the attributes you wanted the first go around. So being able to bind to the epitope you want, have the affinity you want, and manufacturability and developability. And that then kind of started our quest and kind of this new vision of AI drug discovery, and which we started right in 2018.
And around that time, there was sort of this first generation of AI companies coming around. I think most of them were talking about small molecules,
absolutely,
aimed at well-known targets.
Yes,
but in those years, we heard a lot about how we heard a lot of grand claims about AI for drug discovery, but also that there was the data itself was lacking. What was being fed into the AI was either wrong or not enough of it.
Yep.
And you're looking at the developments in AI and saying, well, we have an ability to at least generate a lot of good wet lab data. Absolutely. That we can feed into the AI, and it may point us to some helpful suggestions, I guess, of candidates.
Yeah, and I will be the first to say, right now, AI is we're not at the point where AI can predict biology, but where we're at right now, and Absci has demonstrated this, is the ability to actually design biologics with the attributes you want. Now, why is this important? Well, it gives you the ability to unlock new novel targets that you previously couldn't go after, like GPCRs and ion channels that don't have much surface exposure, so it's difficult for the immune system to generate an antibody towards that. Well, my AI model doesn't care how much surface exposure there is. There just needs to be some surface exposure. And I can then generate antibodies to hit these targets that have been extremely difficult. And this is what's allowing you to kind of go after new novel biology. But it's novel biology based on the design and the ability to hit a target that hadn't been previously hit. Now, where I want this field to go in the future is actually being able to predict the biology itself. What target should we be going after for a particular disease? What epitopes then are, relevant to go after? We're not at that point right now, but we are in this exciting time where we're able to design biologics with the attributes to test hypotheses that we haven't been able to test before.
Mm-hmm. Mm-hmm. So you made this switch around 2018, and then pandemic hits shortly thereafter.
Yes.
And a lot of people get very excited about biotech in general. A lot of money flows into the sector. And so you were positioned to seize the day.
Yeah.
Around that. How about you raised a bunch of money?
Yes.
Became more known.
Yeah.
That you exist up there in the northwestern corner of the United States. How did you navigate that situation?
Yeah, absolutely. So we ended up being able to raise a $65 million Series E that was led by Kasdan Capital and Red Mile in, what was that, 2020. And then it was, shortly thereafter, we raised a crossover round that was 125 million. And then, I don't know, call it 6 months later after that, we were able to then raise, another $220 million through an IPO. And that, really gave us the capital to really build this lab in the loop system that ultimately has kind of led to some of the big breakthroughs that we've had. So we ended up, during that time, being able to acquire two technologies. The first one was Denovium, which brought in our AI capabilities. And, it was really the team that we had acquired that, had some interesting kind of deep learning technology that we thought would be really relevant for the data that we had. And then the second acquisition was Totient, which was based on some reverse immunology platform based on tertiary lymphoid structures. And they were able to show some really exciting results that these tertiary lymphoid structures that are in tumor cells have a very different antibody repertoire than what's in peripheral blood. And if you take those antibodies and you reconstruct them and do a proteome panel screen to find out like what they bind to, and you take them from patients that have had an extraordinary immune response, you can actually discover some new novel targets and biology. And so that was, allowing us to kind of get into target discovery in addition to antibody design. And kind of the AI play that we saw here was that, one of the most time-consuming steps was getting the, after we computationally reconstructed the antibodies from these patients, we had to then go produce the antibodies and then do a proteome panel screen. And that, is a very, time-consuming and expensive step. And we thought, well, if we could kind of go in the reverse direction of our current AI model, where we're going from, target to antibody, we could go antibody to target. You could rapidly de-orphan these antibodies and, discover new novel targets at a very rapid pace. And so that then kind of, those two acquisitions with the capital allowed us to, over the next 4 years, really integrate the AI with the wet lab. And I think that's been the biggest advantage that we've had. We figured out how to be a multilingual organization, how to merge tech with biotech. And this is really the first time, a lot of companies have been able to kind of merge these, two industries together. And we figured out how to create this active learning loop where we can go from data to train our model to then using the same technology, the E. coli and the ACE assay, to then validate the models to see how accurate they are, and being able to do that in a 6-week time period. So we currently have it down to a 6-week active learning cycle, which has just been really instrumental to kind of the success that we've had today within the field.
So that's your lab in a loop. But as you say, we can't yet go from here's an antibody to discovering a target. I'm not really aware of AI discovering new target biology.
We are not there yet.
Yeah.
That is where I want us to be in the future, but we are not there yet.
Okay. But the possibility is
definitely there. Absolutely.
Okay. So you've got these pieces in place that you're assembling. You're taking advantage of, making hay while the sun is shining, so to speak.
Absolutely.
And you hire a bunch of smart people, especially in AI. But now, at some point, all drug discovery companies need to show products and become product engines. Yeah. So where are you at in terms of your pipeline, your product candidates?
Absolutely. So about 2 years ago, we ended up bringing Andreas Busch onto the board. And Andreas was the CSO at Shire. right before the acquisition of Takeda. And prior to that, he was at Bayer. He was the head of R&D there. And Andreas has really an incredible, pedigree. He's gotten over 10 drugs approved under his leadership from bench all the way through approval. And, we brought him on because we wanted a large pharma executive that has built out drug discovery and has done, drug development. And him and I ended up kicking it off. And I ended up, I was like, Andreas, we need to pivot into developing our own pipeline. I really believe in the platform. And ultimately, that's where the value lies, is using these models as a tool to creating the assets. The value isn't necessarily in the AI. It's the value lies in creating differentiated, best in class and first in class therapeutic assets. And I said, step off the board and come build this with me. I need somebody with your experience to go out and build this. And he was newly married, I think a year into his new marriage. And he's like, Uli, his wife is going to kill me. He's like, let me go talk with her and get back to you. And I was like, this isn't going to happen. She's going to be like, hell no. Are you going to go, into this, fast growing AI drug discovery company and go build this out. But sure enough, Uli was supportive, and he ended up stepping off the board, coming on full time. And really that started, 2 years ago, our own internal pipeline development.
Okay. So how do you and Andreas think about setting priorities on what you will do and what you won't do?
Absolutely. So the strategy that we had was the first couple of assets were going to be potential best-in-class assets, where we could show how the AI could create differentiated assets towards targets that had biology that was either known or had been shown to work in the clinic, but we could utilize AI to ultimately meet an unmet medical need and create an ultimate best in class. And that really kind of started the push that we had into TL1A. So
now this was an attempt to validate the technology in the eyes of the scientific community, partners. I remember I saw you at JP Morgan one year, and you were showing me this de novo drug discovery antibodies with the HER2 binder, like Herceptin. So that was a demonstration project.
That was a demonstration showing that you could actually use these models to design an antibody from scratch. And what do I mean by this? Essentially, in the case of HER2, we essentially, took the structure of HER2. We, conditioned the model on the same epitope that HER2 bound to. And then we were able to, redesign the CDRs, in this case, CDRs, heavy chain CDRs 1, 2, and 3, to ultimately bind to the same epitope as HER2. So essentially, being able to design an antibody from scratch
very quickly.
Yeah.
And now you chose not to develop that. There are multiple reasons. Maybe the world doesn't need another HER2 binding antibody. There's plenty of people working on that already.
Yeah.
But it showed what you could do. And then you took those learnings and applied them in another a perceived more green field.
Absolutely. And that's where we then took that and applied it to TL1A. And this particular target is of interest for inflammatory bowel disease and pro-inflammatory pathway. And there had been some recent successes and wins in the space. Prometheus had a great phase 2 readout, which led to the acquisition. Their acquisition or being sold to Merck, and then you had the Roivant deal that also had occurred, as well as the Teva deal. So pharma was definitely very interested in this target.
Those are more recent transactions, but you would have made this decision before that.
We made the decision to go after TL1A before the Merck phase 2 readout.
Yeah.
Yeah.
Now, what was it about TL1 that appeared promising to you?
Yeah, absolutely. I mean, we saw this as a very underappreciated target. Our SVP of immunology, who actually came from Totient, was actually one of the scientists that helped us kind of narrow it down to TL1A, and we just saw it as a very underappreciated target. We believed in the biology, and we saw some of the clinical data that was out there, and we saw, okay, we can leverage our de novo AI platform to create a differentiated antibody that could be a potential best in class in this field. And we're very happy with that ultimate bet that we took.
So you can build off of existing knowledge of biology. In this case, there's quite a bit out there in the public domain about TL1A, and that if you could inhibit this, you would have a pretty positive effect on inflammatory bowel disease.
Absolutely.
And maybe fibrosis?
Yes. Yeah, there's fibrotic indications that you can go after as well.
Okay. So you can sort of follow fast, so to speak.
Absolutely.
Now, how fast? What did you do next?
Absolutely. So this is what's really astonishing, and it's both the AI as well as the team. But from start to finish on this, we were able to get a drug candidate, which we presented at JP Morgan this last year, in 14 months, and we'll be in the clinic within 24 months. Now, to put that in perspective, normally it takes 5.5 years to get an asset into the clinic, and it's usually an investment of $50 to $100 million. And our investment all in from start to finish, and that's completing all of the IND enabling studies, will be roughly $13 to $15 million compared to that $50 to $100 million investment, which is really exciting. You're cutting the timelines by half. And the costs even more than that. And so you get this multiplier effect now. So that $50 to $100 million you normally invest into one asset, you can now invest that into 5 to 7 assets. And we see that as a big game changer.
What kind of advantage do you see your antibody potentially having against the other ones you referenced in the marketplace?
Absolutely. So the first is immunogenicity. If you look at the phase 2 data for Roivant, you see a very high ADA response. I think it's 80% ADA response. Now only 10% of that is neutralizing. Now it was a short trial, and we believe since this is going to be a chronic illness where you're going to likely be taking this medication for the lifetime of the therapy, you're likely going to see those neutralizing antibody responses continue to increase.
You see those ADAs, that's anti-drug antibodies.
Correct.
So the body is trying to fight off, it sees the drug as a little bit foreign.
Absolutely. Yeah. And so your immune system is essentially targeting it. It says, hey, this is a foreign antibody, let's target it, which you do not want with the drugs that you create.
That can cause side effects, or if not side effects, it may at least, reduce the effectiveness.
Yeah, exactly. It could decrease the overall efficacy of it.
So that's the Roivant drug. And the Prometheus, Merck?
Yeah, so the Prometheus showed a lot lower EDA response, but it wasn't as potent as the Roivant molecule. And so what we wanted to do was have a similar or better potency than the Roivant molecule, but have a lower immunogenicity than the Roivant, and and similar immunogenicity profile as the Merck molecule. And when we dove into the data and we did some preclinical testing ourselves. We the hypothesis was that it was B cell mediated. So we looked at T cell activation assays, with both the Roivant and the Merck antibody, and they're pretty similar. And that's what led us to believe that it was complex driven. So it was where the antibody was binding to was ultimately eliciting the immunogenicity response. And the Roivant and Merck molecule is bound in different locations. And so our hypothesis is if we could bind to an adjacent or similar type of epitope as Merck's, we could, have a similar or potentially better immunogenicity response than both Merck and Roivant. And then we also engineered it to have, higher potency than both the Roivant and the Merck molecule. And then the last piece of differentiation, which hasn't been shown clinically yet, but there's some new research that's come out that has led us to believe that there's some, patient populations that actually may not respond to the existing or the competitor molecules that are out there. And so what some of this new body of research is showing is that there are these isoforms of TL1A that patients have. And some of these isoforms, so I guess stepping back, TL1A forms a trimer. And some of these patient populations are showing that they have different isoforms. And these isoforms, some of them actually don't form the trimer. They exist as a monomer, and they're shown to be actually active as monomers. And so if you can actually target both the trimeric state as well as the monomeric state, you could potentially treat a much greater patient population than what's out there. And so if you look at both,
and be able to predict who's going to respond, who enters the trial.
Absolutely. And that's the design aspect that I'm talking about, being able to hone in and target not only the trimer, but also the monomer and all the different isoforms of the monomer, which we've been able to show. And so, again, this shows that you could potentially go after, a broader patient population. You could reduce the immunogenicity and potentially have higher potency, all by being able to design the antibody the right way the first time.
And what about your half-life or dose frequency?
Absolutely. So we have engineered the Fc to have an increased half-life. And our goal is to go from once monthly to once quarterly.
Okay. So you could, and when are you in the clinic now? Or
we will be in the clinic early next year, and then our phase 1 interim readout will be the second half of 25.
Okay, so you will get data a year from now,
yes.
Second half of 25, to confirm whether these hypotheses are right.
Absolutely.
Okay. This is, and you've got more coming behind that, which we don't really have time to talk about. But
absolutely.
What kind of company do you want to really build here for the long term? You're 34 years old. Where do you think this is going to go long term with AI drug discovery?
Absolutely. We want to become the next Regeneron. You look at, what Regeneron, built back in the day. I mean, they were the company that, showed you could, engineer, mice to produce, human antibodies. They humanized the mice. And this is essentially the next iteration of that. Instead of using mice to, generate the antibodies, you're using AI models to design the antibodies and to really unlock new novel biology, shorten the timelines it takes to get into the clinic, and decrease the amount of time or decrease the overall cost that it takes to get these therapies to patients. And so we see this as continuing to grow and ultimately become the next Regeneron. I mean, I think what was built there was phenomenal, and ultimately, at the end of the day, why I get up every morning is to ultimately change patients' lives. I'm passionate about that. I'm passionate about using technology to do that, and so it's this love for technology and the passion to impact patients' lives is why I get up every day and why I fight the good fight. And I truly believe that the design of antibodies is going to completely transform this industry. And we're going to ultimately get to the point where we can use AI to predict biology. We're not there yet. But ultimately, when we unlock that, that's going to start unlocking, personalized medicine. And I mean, I couldn't be more excited about where this industry is headed.
So you might be at a point where you walk before you can run. You start with a third to market, but potentially best in class,
Yes.
TL1A. And maybe that could be your Eylea.
Yes.
And then generates the revenues and cash flows to support all that longer term target discovery biology that you're talking about.
100%. And you have to get that first one right. And we spent a lot of time making sure that TL1A was the right target to go after to really prove out the technology and we firmly believe in the differentiation that this molecule has and the biology. And I think that this is going to be a great proof point for how AI can create differentiated best-in-class antibodies that ultimately make a difference in patients' lives.
Well, that is what it's all about when biotech is at its best.
Absolutely.
Sean McClain, thank you so much for joining me on The Long Run.
This has been great, Luke. Thank you so much.
Thanks for listening to The Long Run, a production of Timmerman Report. Pedro Rosado of Headstepper Media was the sound editor. Music is from D.A. Wallach. See you next episode.